of today is an overview on Theileriosis and Microfilariosis in bovines in India. I've chosen these two topics because they are of importance in the rural economy at the present time. Many of it will be shared during this particular presentation. As all of us know, this livestock scenario is open to us in India because livestock wealth accounts for 1/3 of the agricultural produce. Indian livestock sector contributes substantially to agricultural GDP, and Indian cattle and buffalo population are a great asset augmenting rural economy and national wealth. And here, we need to know and understand that animal health is the foundation to animal production. Now, regarding the economic impact of vector-borne parasitic diseases, we all need to understand that it is a big problem now, especially in the era of climate change. It causes considerable losses to the developing world. The global loss, these are figures which are estimated in different periods of time just to project and make that known about the economic impact of these vector-borne diseases. Global loss due to tick-borne diseases at a particular time was 7 to 18.7 billion US dollars per annum. In India, it was 498.7 million US dollars per annum. In some countries, 80% of the cattle at risk of being infected with tick-borne diseases, and the economic loss here has been estimated to be $248 million per year. Now, coming to the first topic of Theileriosis, tropical bovine Theileriosis is a tick-borne protozoan disease, as all of us know. And currently, in this particular year of 2020, in the OIE list, it is one among the 117 animal diseases. So the importance of it is still very much apparent at this time. It causes major economic constraint on livestock production, and it threatens about 250 million cattle and affects cattle health and productivity. Now here, the mortality at a particular time when crossbreeding program was at its peak was 90% in introduced exotic breeds. And then in indigenous breeds, it was about 5% or less. And coming to the historical perspective, this disease was discovered for the first time and reported by Arnold Theiler, and this genus was later named after him as Theileria. Very soon after his discovery, Shunkowski first described the disease of Theileriosis as a disease entity in the year 1904. Very quickly, within a year, it was reported in India by Lingard, and the annual loss due to tropical Theileriosis in a population of 6.6 million crossbred cows in India during 1989 was around $210 million. This figure has been projected just to make understand that at that time, it was at the peak of the crossbreeding program in India, and this was one of the most important diseases which we were all grappling with and had to deal with. And it became a disease entity with the introduction of crossbreeding program. And the early reports of this disease in indigenous cattle were as early as in 1925 by Edward Cooper, and then by others in Ajwania and Subrailu, Sanan Srinivasan, and Raghvachari. This was an indigenous cattle, and only later on, when in the 1970s, we found this to be a big problem when crossbreeding program came in. So it became a disease entity, and it was reported one by one from different states of India. This is just a gist to show the importance of that. Now, coming to the geographical distribution of this disease. Now, among the three important species, as you can see here, Theileria annulata, which is dark colored here, puts 250 million cattle at risk. Very widespread and important. Theileria parva, although it's a very severe disease, it affects only 50 million cattle at risk, and it is restricted to a small zone. Theileria buffali orientalis or sargentii. This is a group known as such, is relatively benign, but it is very widespread, as you can see, all over in this particular map, covering most of the countries. Now, the different species of Theileria and distribution I wish to go through just to highlight where it occurs and how it is, and what are the vectors which are distributed and which are transmitting this infection. Theileria annulata, which causes tropical bovine theileriosis, has Hyaloma species as its vector, which is very widespread in India. Pathogenicity is quite high. And as you can see, the distribution is also very wide, right from North Africa up to Southern Europe, with various parts of Asia, Middle East, and the Indian subcontinent. Cattle and Asian buffalo act as the hosts, definitive hosts. Theileria parva, for which Rhipicephalus appendiculatus and Rhipicephalus zambiziensis are the vectors, has a high pathogenicity, but it is restricted only to eastern, central, and southern parts of Africa, where cattle and African buffaloes are affected. Theileria mutans, for which amblyoma, the tick vectors, are low to non-pathogenic, and it exists only in the sub-Saharan Africa, where cattle and African buffaloes act as vectors. These are the other species. Now, among these, as we can see, they're all from other countries. We have the second species which occurs in India very commonly, which is Theileria buffali sargentia orientalis, and for which Haemaphysalis ticks act as vectors. Different species of Haemaphysalis are there, out of which these are the common ones, longicornis and punctata, and Rhipicephalus microplus, which was formerly called as Buplus microplus, is moderately to highly pathogenic under high stress. It is cosmopolitan, widely distributed, and it has caused disease outbreaks in the recent times, as we have all understood. I'll explain it to you a little later. It affects both cattle and Asian buffaloes. And the species, the other species are Theileria taurotragai, which occurs in cattle and eland antelopes in southern and eastern Africa. Now here, I would like to just, although my topic is restricted to bovines, just a small bit which I would like to project here is about sheep and goats being affected by different species. This is just to highlight the emergence of important species which are causing high pathogenicity. Formerly, we understood that Theileria hirsi, now called as Lesterquadai, was causing very high pathogenicity in sheep and goats. But very recently, in the recent years, we have come across outbreaks and mortality and morbidity due to a species that is Theileria leuvenscheni, which was transmitted by Haemaphysalis quincaiensis. But here in India, it's transmitted by longicornis and cachensis. And we did a study in Bangalore for the first time and found it to be highly pathogenic. And this was the new vector which was identified, Haemaphysalis cachensis. So it's an emerging disease at this time. And as we know, it has come, when we did the phylogenetic and molecular studies, we understood that it has come from China. And then the other species which occur in sheep and goats are the less pathogenic ones like Ovis, Recondita, and Separata, just to highlight the importance of emerging species among the small ruminants. Now, the definitive hosts, as we all know, for tropical bovine theileriosis are cattle and buffaloes. Although buffaloes carry the disease, they don't manifest it as much as the crossbred and the exotic cattle. Now, epidemiologically, tropical bovine theileriosis is mostly observed in summer and rainy seasons. And here, the tick population so happens to coincide during this particular season because the ticks favor the animals which are confined, huddled, and kept in confinement. So it's very ideal for them to breed, and that's how they transmit the disease. And this disease is influenced by stress, hot and humid climate. Indigenous cattle are affected in the sense they carry it, but they don't cause any serious disease in them. But the exotic cattle are highly susceptible. For example, when we import animals for crossbreeding program, it becomes a big issue which we need to keep in mind. And buffaloes are resistant than cattle. Now, this transmission of this tropical bovine theileriosis is caused by the main longirostrate tick, Hyaloma anatolicum aletolicum, which is widespread throughout India. And it is a three-host tick, which makes it very ideal for the disease transmission. And here, the transmission is by stage to stage, and mechanical transmission could also occur by tick bite and inoculation. Ticks remain infected on pasture for 2 years, which makes it very ideal to be carried on for a long time. in a particular endemic spot, and Theileria sporozoid transmitted saliva of ticks to the definitive host is the main method of transmission. The parasites persist faster in hot climate and in nymphs compared with adults. So in this regard, just a picture of the tree host tick life cycle, just to explain the importance of transmission and biology. Now, when the larva picks it up, here it gets developed in the nymphal stage, and as the nymph is about to molt, it transmits the disease to the next host, and it remains with the adult for a very long time. Both nymphs and adults carry it for a long time, and this is how it is propagated in nature in endemic spots. Now, when coming to the life cycle, this is all well understood by this time, but the idea of me bringing this back is to make us known that the life cycle of Theileria was not known for a very long time. It was thought to be only asexual, and even in the book Soulsby, which we all used to refer as a very important book for us, the life cycle was not explained previously. But later on, in the past 2 or 3 decades, Eulenberg, Theiler, and this team, Melhorn, all of them joined together, German scientists especially, and found that it has a sexual life cycle. In this regard, we can see here, right from the transmission from ticks, which transmitted, the development of it occurs in the gut of the tick. The stages, the sporozoites migrate to the salivary gland, and when the tick bites, it transmits the disease. It goes to become the tropozoid, schizont, and here, very importantly, clonal replication of infected cells take place. And this leads to a lot of pathogenicity, as we'll see later, and then it differentiates into merozoites and then goes into the erythrocytes as pyroplasms. So here we have these 3 stages: the macroschizonts found in the lymphocytes, the merozoites which differentiate and come into the blood in the pyroplasm form, which is seen in the infected erythrocytes. So that was the life cycle of Theileria annulata. Now, oriental theileriosis, which was also known as Asian theileriosis or Theileria associated bovine anemia. These are the 3 terms commonly used. It's caused by Theileria orientalis or buffali, and this group was first identified in Australian cattle as early as 1910, but it still persists to this day, causing a lot of problem in this country itself. Early reports from Chennai, Maharashtra, and Karnataka were based on morphology and benign disease. See, we all know that Theileria is pleomorphic, and it's not possible to speciate based on morphology. They mainly occur as ring forms, round, and even rods, and therefore we can't identify them. And in this case, Theileria orientalis appears as a bar-like rectangular structure which looks like a refractile bodies in wet blood mounts. So this gave a lot of speculation, and people started using this method to be able to identify and differentiate. Then it is associated with a bar or veil-like structure along with a pyroplasm, which can be seen only in wet mounts. It cannot be easily visualized in stained blood smears because this veil-like covering gets masked in the stain. And there are very few schizonts in the lymphocytes. This parasite is transmitted by Haemaphysalis bispinosa ticks to cattle and Rhipicephalus microprus to cattle and water buffalo. In this case, also transstadial transmission occurs. Now, regarding Theileria orientalis, there are variations in the major pyroplasm surface protein, which has been able to be known by molecular biology techniques. And this gene has been used to classify members of the orientalis group. It's called a group because 11 genotypes are known. In India, 3 types, 1, 3, and 7 are found in cattle, and only type N2 is found in water buffaloes. In Kerala, in recent studies, the MPSP genotype, the major pyroplasm surface protein gene genotype, N2 was reported to cause clinical theileriosis in Asian water buffaloes, and the parasite now is globally spread. But countries which are impacted by clinical theileriosis, oriental theileriosis, include Australia, New Zealand, Japan, Korea, China, and Vietnam. And now we have a complete genome of Theileria orientalis, the ICEDA, which has now been sequenced and annotated and is available for further research. Now, clinical theileriosis, like I just said, of the orientalis type, has been linked to pathogenic genotype of the parasite ICEDA, which is the type 2. Clinical outbreaks of Theileria orientalis have been increasingly observed throughout Asia and Australasia. In early stages of clinical oriental theileriosis, signs of muscle weakness, ataxia, and abortion have been observed in infected animals. A variety of clinical findings such as lack of appetite, pyrexia, elevated heart rate, abnormal breathing, pale mucous membranes, and jaundice have been reported. Now, we have to accept and understand that oriental theileriosis is endemic in India. It is a non-transforming strain exerting its major pathogenic effects via erythrocyte destruction. So here, the mechanism of pathogenicity is different from Theileria annulata because here it all occurs in the erythrocyte, unlike in Theileria annulata, which all occurs in the lymphocytes. Theileria orientalis or buffali invade and destroy erythrocytes, and the main resulting changes are anemic and icteric changes. Most of the time it causes a benign disease, but can cause clinical disease and mortality under stress and exposure to naive animals. For example, when we introduce carrier animals into places where this has not been exposed to those particular population there, it leads to the clinical disease. So now coming to pathogenesis of tropical theileriosis, which is by this time well understood. The incubation period is 15 to 25 days, right from the time of tick bite, the sporozoite, which enters through the skin into the blood, first goes to the closest lymph gland, lymphoblast cells, get spread throughout the body to all the lymphocytes and become schizons. It leads to hyperplasia of lymph gland, then it gets transformed to merozoites, and there is destruction of RBC and anemia. But the main pathogenicity is due to the lymphocyte destruction and proliferation. Now, going into the mechanisms of pathogenesis, Theileria annulata schizons induce proliferation of the host cell in synchrony with their own replication, creating clones of parasite-infected cells. So the parasite is able to manipulate the immune response, and to such an extent that the presence of parasites in the pituitary gland and their presence and extensive damage in the adrenal glands indicate that Theileria annulata disrupts the regulation of the immune and endocrine systems, including temperature regulation, feeding, growth, and nutrient utilization. Theileria annulata is lymphoproliferative in nature. So now coming to the gist of the clinical signs, varied clinical signs are seen. The first sign will be increase in temperature, body temperature. Then second very indicative sign will be the enlargement of superficial lymph nodes, which enlarge to a very great extent, making them very palpable. And then we see other changes such as watery nasal discharge, subconjunctival hemorrhages, and then we have changes in the eye like chemosis, exophthalmosis, which could be unilateral exophthalmosis or bilateral, as you can see in these pictures. And then various symptoms, because all the tracts are affected. Even the gastrointestinal tract, the lymphoid system is affected. Therefore, the lymphocytes, the Peyer's patches, and so on. So diarrhea with mucus and blood clots are an indicative sign. Then we have other signs like pseudopericarditis, where we see a string-like jugular vein, very prominent here, and skin lesions. Then we also have other signs like corneal opacity and the highlighted post-mortem lesions, which we see initially will be splenomegaly, and then when we cut across and see the spleen, there will be atrophy because of depletion. And then we have enlarged lymph nodes, very abnormally enlarged. When you cut open, you can see them. And then we have other signs like very indicative and specific signs like necrotic foci in the abomasal punched ulcers. We always have a common question to ask in our exams to students. What are the indicative signs? And we always say abomasal ulcers. But many don't understand why it happens, why it should happen like that. It is because of degeneration and necrosis of lymphoid patches in the abomasal. And then we have white nodule lymphocytic infiltration. And then we have swollen edematous lungs, emphysema. Then we have changes in the hemorrhages in the endocardium We see ecchymotic hemorrhages on the epicardium and endocardium of heart. So these are all the indicative important signs which are caused because of the widespread lymphocytic proliferation. Diagnosis, very important because it's not as easy as we put it down. Now, in diagnosis, we first take a history. What we mean by this is history will be contact and a tick bite. Many times we look for a large number of ticks on the body. In the case of Theileriosis and babesiosis, but we need to understand that there needn't be many ticks. A single tick, single nymph is loaded with sporozoites. So it can very nicely spread the disease, but a higher number of ticks can cause a full-blown disease within a short time. So history of tick bite. Clinical signs, like I just showed, fever, and then enlarged lymph nodes and the others, and about the mucous membrane of the eye and conjunctiva and so on. Microscopic examination of stained blood smears is the most commonly done. It's a pen side or diagnosis, which can be done fast or in a small diagnostic laboratory, where the pyroplasms are seen. They're very small. They're less than 2.5 microns. They're ovoid, annular, ring or rod shaped. And this is what makes it difficult to speciate. And then lymph node biopsy smears are taken and we make a stain and look for Cox blue bodies. Now, there are a number of serological tests like the immunofluorescent antibody technique and the ELISA. And later, we got the molecular tests like the polymerase chain reaction and LAMP. Now, going into the pros and cons of these various diagnostic tests, we have blood smear. Here we can see, typically we see the signet ring shaped bodies, the dot-like bodies, the ring-like, annular, rod-like. So this makes it a little difficult to correctly identify, and it requires expertise and practice. Cox blue bodies, which we see in lymph node smears showing the schizons. Now, coming to the limitations of blood smear examination. Now, whenever we take a blood smear from an animal, from an endemic area, they are bound to see a few parasites in that. Can we come to a conclusion? Because these long-lasting carrier state makes it very difficult to say whether it's a clinical, subclinical, or exposed state. Theileria pyroplasms are difficult to detect due to small size and stained blood smears, and discrimination of Theileria annulata from other species is a problem. Like, for example, now here in India, we have two species, common, Theileria annulata and orientalis. So we need to get to which is the correct parasite species. And now sometimes mixed infections are there. If both occur, what do we do? Then we have smear method, which could have technical problems, like wrong blood smear preparations are done by field veterinarians many times due to difficulties. Then we come to a wrong diagnosis because we can't see properly, can't stain properly, and they're not in a proper state. Low sensitivity in diagnosis in carrier cows. The smear should be taken at the height of temperature. And then some smears are very difficult to stain and detect. It's labor intensive and limitation is there for large scale survey. Now, the alternative here will be a serological test, which can be done only in a proper well-equipped laboratory. Immunofluorescent test, antibody test is one of the good tests for many diseases because it gives a clear fluorescence in this reaction. That's what is given in these steps. It requires different steps. It requires specialization, and it requires so many inputs. And here we need a good microscope, fluorescent microscope specifically, and it's fairly specific, easy to perform, but it's sensitive. The disadvantages could be cross-reactivity, limitation for large-scale survey, and cost of producing specific purified antigens, which is very high. Now, the next other test which came into being, which was widely used, is ELISA. ELISA does have its own strengths. Different types of ELISA are there. We have done, for example, you could do a simple test like dot ELISA if it is standardized. It's quick, easy to do. We have done it and found it to be quite reliable. And then we have plate ELISA, which is a conventional type, which gives a clear picture, and you can even do a quantitative estimation of the antibodies. And then one more important thing about ELISA is it can be used in seroepidemiology, and it can also monitor induced immunity in vaccinated animals. We did an avidin biotin ELISA of vaccinated animals in Karnataka and found it to be very reliable in detecting the protective state of vaccinated cows born to vaccinated animals. And it has less operator error, and only disadvantage is it is cross-reactive because of the type of antigen which we use. Now, next we come to molecular methods which are very reliable and the in thing for the day. Now, molecular methods for correct identification and detection is what we rely on. Right now, we are groping with this pandemic, and then we go for a simple antigen test. Yesterday, there was in the news about certain quick test, which was brought in by one of the companies. Now, these quick tests are indicative, but it's not reliable completely. We need to do an RT-PCR. So like that, PCR is the ultimate thing which gives a confirmation, and it gives a very correct diagnosis, specific and sensitive diagnosis. As for the other diseases with Theileria annulate also, many studies have been done throughout India and in the world, and unique DNA sequences provide very high levels of specificity for the diagnosis and identification of parasite species and strains. For example, when the disease was in its peak in India, in different parts of India, a lot of work was done throughout. It was done right from Uttar Pradesh, it was done in Hisar, then we did it in Chennai, Maharashtra, and various places, including South India, in Chennai and in Karnataka. And at that time, there was no PCR, but we did it with the other tests. And what I would like to say is a lot of strains were found. Strains were found, Hisar strain, Bangalore strain, Chennai strain, so many strains. And then there was a debate when a vaccine came in, whether that single vaccine will be effective against all the strains. Fortunately, it was. So this is important where we can see the species and strains, and it gives specific and sensitive diagnosis for Theileria annulata. Carriers can also be detected by PCR. And then now we have a simpler version of molecular method called the loop-mediated isothermal amplification, called LAMP. It is sensitive and less time consuming, more simpler than PCR. It is an isothermal technique which uses a water bath, and it has been used widely and usefully for Babesia and Theileria, and it can be used without DNA extraction. Now, the advantages of both these tests, PCR and the other molecular tests, like I just told you, it is rapid, high sensitivity and specificity, and carriers are detected, very useful in molecular epidemiology, less laborious, and it can be used for a variety of clinical samples. Now, direct analysis. Now, what we mean by less laborious is it should be standardized. It should be in a proper laboratory. It cannot be done everywhere and anywhere. Once it is done, it is okay. Then we have direct analysis of clinical samples and superiority in separating parasitic infection associated with clinical signs, whether it's the clinical form or subclinical form. Now, after knowing about the diagnosis, we come to chemotherapy. By now, we are very fortunate in an age where drugs are available for many of the parasitic diseases, not only in the world, it's found in India as well. Because of this World Trade uh, uh, Organization which came into being, we have access to drugs and various things. And therefore, earlier it was done so elsewhere. We tried a few trials in the beginning from Cooper's. It was at that time, it was newly introduced about 30 to 40 years ago. And these two drugs became specific for the treatment of theileriosis. Parocone and buparocone, they are hydroxynaphthoquinone compounds. They are active against both the schizont and pyroplasm stages. Antithilarial activity was detected in vitro for the first time, then it was tried in vivo. It has a long plasma half-life, low toxicity, and the mode of action is it interferes with electron transport within mitochondria at the ubiquinone level, as is explained in this diagram. Now, the dosage is 2.5 milligram per kg body weight intramuscularly as a 5% solution. It affects the pyroplasms as well and changes the morphology, showing that it is acting. But here we need to see in bovines, milk withholding period is 2 days, and the cattle should not be slaughtered for human consumption within 42 days of treatment. Now, sorry. Now, for early disease treatment and prophylaxis, we have long-acting oxytetracyclines, which arrest development of parasites and lymphocytes, and thus reduce the rate of invasion of cells. So if you can arrest it at the early stage, as soon as fever is seen, we can cut down the morbidity to a great extent. Now, the dosages of oxytetracycline are 10 milligram per kg per day to 15 milligram per kg per day, 3 days. Only thing is, it's a very viscous thing, oily preparation. It may cause a lot of pain and swelling at the site. So we need to be careful with that. And then long-acting tetracycline, which came into production a little later on, 20 milligram per kg intramuscularly, given 1 or 2 times, depending upon the thing. It was also very effective. But only thing we need to remember is this only helps in the early stage of the disease, and it arrests schizogony. The other drugs which have been available and which have been researched upon throughout many countries are imidocarb dipropionate and primaquine diphosphate, which have been shown to be effective in reducing Theileria orientalis parasitemia in cattle at doses of 1.2 milligram per kg body weight and 1 milligram per kg body weight respectively. So now this particular the regimen needs to be followed because we need to, when Theileria orientalis becomes a disease problem, we have to go to these drugs. And then diminazine aceturate, which is commonly used in babesiosis, leads only to partial recovery and is not recommended as such. Now here we conclude about the treatment regarding theileriosis that the most active anti-theileриal compound in the hydroxynaphthoquinone series is buparocone, and also the quinazolinone derivative halofuginine lactate. Both drugs are effective in treating early stages of Theileria annulata infection. Now, this was a report which just shows between parocone and buparocone the efficacy. Buparocone scores high and it is effective, and the dosage already explained. And so this is one of the drugs which has come in, which should be given in a reasonably early time when the morbidity and pathogenicity has not increased greatly. Only then we can save the animal and also cure the disease. Prophylactic use of Schizon tissue culture, vaccine, and chemotherapy with buparocone could be one of the most promising means of controlling theileriosis. Incidentally, now we are in an era when ethno-veterinary practices are being propagated and also strengthened because of so many difficulties in the field level and also alternate therapies to be seen. Incidentally, just a mention of a herbal compound called Paganum harmala, a picture of which is given here. The seed extract was found to be pharmacologically active, containing several alkaloids, and the dose in these 2 reports of by scientists showed that 5 milligram per kg body weight intramuscularly once for 5 days showed a recovery rate of 78% and 93% in another study. Now, supportive therapy forms very important part in a clinical management of theileriosis, where we need to give liver tonics, hematonics, and also sometimes blood transfusion if the hemoglobin level is 5 gram per DL, and vitamin supplementations with B12 and B complex. How do we control this disease? To restrain the biotic potential of the parasite and the vector needs to be worked hand in hand. Otherwise, we can't control this disease. We need to, that's why the life cycle was highlighted to say where we can cut it down. And it can be achieved by improving management, destroying vectors, preventive dose of drugs, epidemiological studies, and immunoprophylaxis. And then tick control, we all know, I won't deal with it in great detail here because it's a subject by itself. It can be done in various ways. The more newer methods I'd like to highlight on regarding control of ticks. We need to understand and cut the life cycle in the environment by these various methods. And also when it goes beyond control in farm conditions, we need to use dips and sprays. Now, the other methods could be predators, repellent plants, which are abundantly available in India. Only thing they need to be promoted and cultivated around livestock farms and pastures for tick control. I'd like to highlight that Stylosanthus scabra, one of the leguminous fodders, which is widely found in various parts, has tick trapping properties. and it's very safe. Being a leguminous fodder, it can be used and cultivated. Seeds are available in India all throughout, and it can help in tick control in the farm. Chemosterilization with different methods, which are listed here. Sterile males, they're all a little academic, but semiochemical approach is one of the newer approaches which has been evaluated in various studies in Chennai and elsewhere and found to be very promising. It uses a pheromone-based product along with an acaricide, and it kills the ticks, and it can be used in various ways in the farms, and it can control the disease. So here I've just explained to you about the different methods, and this is the pheromone impregnated decoys for attracting and killing ticks, which is called the semiochemical method. And now we have also tried many biological agents and vaccines for ticks, but unfortunately we have only one vaccine for the tick, Bacillus microplus. And then we have disadvantages of acaricidal control, so we need to keep this in mind when we use chemicals. And now coming to the last part of the immunoprophylaxis, little bit of highlight. Actually, for a long time, and this disease was widespread, attenuation was tried for and demonstrated for the first time in Israel, and then the Schizons were attenuated, and it reduces significantly the incidence of theileriosis from 85 to 100%. And this is later on, this technology was taken, and we found our own vaccine in India, which was evaluated, and then it was, I'm sorry, it was patented, widely available, and it needs to be propagated. Actually, here in the Karnataka Milk Federation, this vaccine is available, given to farmers, and it has reduced the incidence of of this disease in vaccinated animals. It can be used widely. I'm sure all of you know about the various properties of this vaccine, and also it needs to be kept in liquid nitrogen. Now, one-time vaccination in field conditions for the adult animals will do. And then a booster, what happens is in endemic places, there is a booster because of continued infection with tick bites. And young animals need to be vaccinated because protective immunity from the mother, vaccinated mothers, is short-lived. Therefore, it needs to be given to young calves so that they will not get into a full-blown disease. Calves of vaccinated dam are also susceptible and need vaccination. So that was about theileriosis in bovines. And coming to the next topic, which I'd like to deal, which is not well known and understood, and that's why I thought I'll put it into this presentation in this webinar, because phylarid nematodes are very widespread in India. In different parts, we have different phylarids. And then the adult phylarid nematodes inhabit the skin and connective tissue and ligamentum nuchae and so on. List of them is here. What I'd like to say is, see this Paraphylaria and Elaphora, Paraphylaria, Stephanophylaria, and Onchocerca gibsoni and gutturosa. These species are found in the skin or connective tissue or ligamentum nuchae. They don't shed the microfilaria into the blood. So we need to, whenever the disease occurs, it occurs in localized in the skin as dermatitis, hump sore, and so on. So we need to take skin scrapings and look for microfilaria. Only in the case of Ceteria species, Labiatopapillosa, digitata, cervi, and Onchocerca armulata. These are the only 4 parasites which shed their microfilaria into the blood. And in this, Ceterial microfilariosis causes a clinical syndrome. Phylarid nematodes can cause significant morbidity leading to economic losses. Mortality has also been reported in severe untreated cases. For example, in high-yielding animals, we have lost in Karnataka because of this particular parasite. And then microfilariosis caused by Ceteria sometimes remains undetected. And one thing which you need to know is it's widespread because mosquitoes act as the intermediate hosts. The adult worms dwell in the peritoneal cavity of herbivores. The sheathed microfilaria circulate in the peripheral blood and cause clinical disease. Now, microfilariosis in bovines, like I just told you, are caused by the larval stages of Ceteria and Onchocerca marmulata, but more important, Ceteria. What happens is the L1 are introduced, are picked up from the blood or peripheral blood of infected cattle and buffaloes. It causes disease in both cattle and buffaloes. Here, this is one important thing which we need to understand. It causes systemic disease known as microfilariosis. The mosquito species Aedes aegypti, albopictus, pembamiensis, Culex sapiens, Anopheles species, and Culicoides also act as vectors, which are widespread in India, and that's why this disease also is widespread. The life cycle is very simple here, as we can see. In the peritoneal cavity, we have the setaria. They release the motile embryos, L1 microfilaria, into the peripheral circulation, and these are picked up when the mosquito bites. And in the mosquito, L2 and L3 stages develop in 2 to 3 weeks, and when the mosquito bites another infected host, it's released into the system and becomes an adult one. So this is the life cycle, and which I like to highlight. Microfilariosis in buffaloes was reported as early as 1972 as a clinical disease from the Tarai region of Kumaon Hills in Pantnagar, later on in many other places in parts of India. This was just to highlight the very many reports right from Balerao up to Sharma of UP. And in this regard, all these reports are there even from Maharashtra. We have had studies, Andhra Pradesh, Karnataka. Now here I'd like to just draw your attention to one study of Mohiuddin in 1955 in Karnataka. At that particular time, bovine paraplegia was a big problem in some parts of Karnataka. And the animals used to become paraplegic, and the etiology was tried to be understood so that control can be made. Different etiologies were looked into, like nutritional, it was looked into fungal, viral, bacterial, and so on. But they couldn't zero down on anything. But this Dr. Mohiuddin, who happened to be our teacher at that time, he did this study and found in one histopathological section of the spinal cord, he found the larvae of Setaria, and that is why it was proven that microfilariosis, cerebrospinal microfilariosis, is an important disease entity which is prevalent even now. Now, prevalence of microfilariosis in buffaloes is prevalent throughout the year. The incidence varies from 5.2 to 26.06 in these two studies. The high incidence is seen in the months of July, August, September, October, November. During the rainy seasons, adult animals are more commonly affected. High-yielding cows are affected more, especially the high milch breeds. Murrah cross buffaloes are found to be more commonly affected than the other breeds, and it's more important as a disease syndrome in the early lactation period. Now, a number of studies were conducted. Actually, field veterinarians from Andhra Pradesh were the first ones to observe this disease and report it in those days. Later on, many studies were done in various universities and colleges in Uttar Pradesh, Orissa, Madhya Pradesh, Karnataka, and Tamil Nadu, and this disease was got to be known in a better way regarding its prevalence state as well as treatment and control. The prevalence was found to be extremely high in coastal areas of Andhra Pradesh and in the hilly and forest areas of Tarai region of Kumaon Hills and Pantnagar, more commonly found in buffaloes than cattle. Now here, a plethora of clinical symptoms. It's very difficult to highlight and pinpoint which is the indicative symptom. That's why I put this up in big letters to just highlight weakness, emaciation, varying degrees of loss of appetite, chronic indigestion, mucopurulent exudate from nostrils, dyspnea, cough, conjunction of conjunctival mucosae, scleral capillaries, dry muzzle, rough skin coat, pityriasis, shrunken eyeball, epiphora, depression at the temporal fossa, impaction of rumen, drop in milk yield, lameness, swelling of joints, and udder edema. So you can see very difficult to pinpoint because of these plethora of clinical symptoms. Sometimes nervous symptoms can also occur, dashing against hard objects and so on. So all the symptoms fluctuate throughout the day. One important thing about this microfilariosis, during the daytime, there's not much of symptoms indicated, more pronounced in the evening and late hours. So whenever we suspect this disease, animals show a high increased pulse, respiration, fever in the night. Therefore, we ask veterinarians and farmers to get the blood collected in the evening hours so that we can correctly identify. Otherwise, it's difficult to see the microfilaria by the different methods. Some animals exhibit nervous signs like striking the head against wall and constant scratching, Gastrointestinal disorders, swelling of joints have also been noticed in various studies, including our study in Bangalore, and erratic migration of Ceteria labiatopapillosa larvae in cattle in these so many animals, large and small ruminants, in the CNS can cause lumbar paralysis, which is irreversible, fatal, and causes cerebrospinal nematodosis. And erratic migration um, could cause even ocular or urinary theileriosis. Now, the blood picture, clinical pathology, and biochemistry has been studied, and various changes have been found, which are described here, like decreased hemoglobin, PCV, erythrocyte count, leading to, resembling a macrocytic hypochronic anemia. Mild anisocytosis, moderate increase in reticulocyte count is observed. Relative leukocytosis associated with neutropenia, lymphocytosis, monocytopenia, and eosinophilia are other changes observed. Now, in the biochemical changes, we see increase in total serum protein and globulin, decrease in serum albumin and albumin globulin ratio, and hyperproteinemia with significant increase in plasma fibrinogen has also been reported. Reduced blood glucose levels, where concentration of total serum lipids and serum enzyme activities have been found to be significantly increased. Now, how do we diagnose this disease? Actually, we did a number of tests. The widely used method is the modified Knott's method. Very simple method to do. Field-oriented method can be done in an ordinary laboratory. Although it has 89.79% sensitivity, it is the easy and good method which can be done. And we tried another method which was looked into from human filariosis, called the citrate saponin acid method. It requires a few inputs, but it is a reliable method. It can be done in a laboratory where we find these reagents. It's a good method. Now, direct wet smear method has a sensitivity of 65.30%, and GIMSA stained blood smear has the lowest sensitivity of 59.18%. So modified Knott's method is moderately a good method, easy method, field-oriented method, can be practiced widely and reliably. The characteristic features of microfilaria are more clear when 0.5 methylene blue and GIMSA stains were used for staining in one of our studies. Now, these are some pictures just to show how they look like in the direct wet mount method and citrate saponin acid method, both methods. We find these larvae, all the larvae in large numbers. In wet mount, in modified Knott's method, even if there's a single microfilaria, we can see it. That is the advantage of this method. Now, when we do a GIMSA staining, we see this type of staining, very clear and easy to identify. Now, under oil immersion lens, we can see the Ceteria very clearly like this, sheathed, and we can study the morphology very easily and identify it correctly in a parasitological method. Now, coming to the treatment and control of microfilariosis, sectarial microfilariosis at both cattle and buffaloes can be successfully treated with ivermectin and doramectin. Actually, these drugs came in later. Therefore, a number of studies which were done previously, different drugs were tried and evaluated without much benefit. But once ivermectin came in, and later on doramectin, the other form, we found it to be very effective in our study, and it continues to be useful. And then this is the report of a study which was done in Parbani by a clinical case, which I will show you just next. In acute nervous type, 3 doses of injection of ivermectin at the same 200 microgram per kg body weight, subcutaneous at weekly intervals, along with supportive therapy with non-steroidal anti-inflammatory drugs, fluid therapy, antihistaminics, and hematinic preparation for 5 days proved to be very effective in treating a nondescript animal of this disease. Mosquito control forms a very important part with bed nets, repellents, and biopesticides. As we all know, mosquito control is easy said than done. We are groping with malaria, dengue, and so many diseases. Mosquito control is a big challenge. Now, I'd like to, that may look hypothetical, but I'd like to tell you that I have seen, and it is a fact, that barriers, physical barriers using bed nets are used for high-yielding animals and in farms. This is a very practical, useful, and good method of mosquito control. And the use of repellents and biopesticides is another method which needs to be taken care of. Number of biopesticides are available and they can all be very successfully used, neem-based, eucalyptus-based, and various other components like the oil of citronella, lemongrass oil, and the others. Now, just to make it very clinically sound, I have brought this case report, which was described in one of the papers by a clinical case which was dealt by one of the scientists, Dr. Satish Digraskar and his team in, in Parbani. This is a case of an acute microfilariosis with nervous symptoms, which was reported in a 4.5-year-old nondescript cow. There was complete hemorrhagia, salivation, stiff gait, convulsion since 8 to 10 days, nervous symptoms. So on clinical examination, moderate dehydration, rough hair coat, congested mucous membrane, but the temperature was normal because this was done during the day. Increased respiration and heart rate was observed. Now, if you had done a test during the night, you would have found high body temperature also. But this was done during the clinical hours, and a wet blood film and modified Knott's method examination revealed microfilaria of Ceteria, and the hematological examination revealed decreased hemoglobin, PCV, and total erythrocyte count. And on the basis of symptoms and laboratory examination, this case was confirmed as microfilariosis, and the cow was successfully treated as above. So this was just to highlight the importance of clinical microfilariosis in bovines, especially buffaloes, which needs to be taken care of in day-to-day practice.